Homeostatic regulation.
In this video, we will explain the homeostatic processes that regulate glucose levels. Homeostasis is the body's mechanism of maintaining steady-state conditions in an open system. When changes occur to the body, homeostatic mechanisms work together in organized self-government to oppose those changes. This helps ensure our bodies are able to function properly.
When changes in blood glucose levels occur, homeostatic mechanisms take action to return the blood glucose levels to normal. When glucose levels get too high, the body must lower them by increasing cellular uptake of glucose. When glucose levels get too low, the body must increase blood sugar levels to get back to normal. Next, we will go more in depth for understanding of these topics. Plasma glucose comes from three sources: intestinal absorption during the fed state, glycogenolysis, and gluconeogenesis. The processes of glycogenolysis and gluconeogenesis will be discussed further later.
Hyperglycemia is when blood sugar levels are abnormally high. To prevent this, the body takes action when blood sugar levels are above the range of 70 to 110 milligrams per liter. When this occurs, beta cells in the pancreas detect that the plasma glucose levels are too high and increase secretion of insulin. Insulin is an anabolic hormone that lowers glucose levels three ways. The first way is that it causes cells to increase glucose uptake. The second way is that it increases glycogenesis in the liver. And the third way is that it inhibits glucagon secretion.
Insulin increases glucose uptake by binding to tyrosine kinase receptors on cells, which causes a phosphorylation cascade, leading to the exocytosis of vesicles containing GLUT. GLUT4 proteins are transport channel proteins that allow glucose to passively diffuse into the cell. As these proteins become part of the membrane, they increase glucose uptake by cells.
Insulin also increases glycogenesis in the liver and muscle cells. Essentially, through a complex biochemical pathway, glucose is converted into glycogen. Glycogen is the major storage carbohydrate of animals, much like starch is in plants. Insulin also decreases glucagon secretion. We will discuss glucagon in more detail next.
Hypoglycemia is when the blood sugar is abnormally low. To prevent this, the body takes action when glucose levels fall below the range of 70 to 110 milligrams per deciliter. When this occurs, alpha cells in the pancreas detect that the plasma glucose levels are too low and increase secretion of glucagon. Glucagon is a catabolic hormone that helps increase plasma glucose levels by increasing glycogenolysis and increasing glyconeogenesis in the liver, and also increasing lipolysis.
Glucagon increases plasma glucose levels by signaling the liver to begin three processes which produces glucose. The first is glycogenolysis. This is the process of breaking down glycogen stores into glucose. Next is lipolysis, where triglycerides are broken down into glycerol, which then becomes glucose. Finally, the liver utilizes amino acids for gluconeogenesis, or the synthesis of glucose. The produced glucose is then propagated throughout the body to be used as an energy source. These are the summaries of the processes using negative feedback loops. When blood sugar rises, the beta cells of the pancreas are stimulated and release insulin. Insulin goes on to stimulate adipose, muscle, and liver cells. Muscle and liver cells initiate glycogenesis, and all three of the cells uptake glucose in response to insulin stimulation. The result is a decrease in blood sugar or blood glucose. When blood sugar is low, alpha cells in the pancreas are stimulated and release glucagon. Glucagon also stimulates liver, adipose, and muscle cells. The overall effect from glucagon is an increase in glycogenolysis, lipolysis, and gluconeogenesis. The net result from this effect is an increase in blood sugar or blood glucose.
There are two types of diabetes mellitus, but we will be focusing on the effects of type 2 diabetes in this video. In type 2 diabetes, cells become resistant to the effects of insulin, meaning glucose builds up in the plasma and isn't absorbed by the cells as quickly. This means the body needs to produce a lot more insulin than it would normally, which is bad for beta cells in the pancreas. While type 2 diabetes can be managed by exercise and a healthy diet, oftentimes it results in patients having to take supplementary insulin.
Why is this bad? The increase of glucose in the bloodstream causes an increase of water into the urine as kidneys try to filter out excess glucose, dehydrating the person. Since cells are unable to access glucose, the person may always feel hungry. Long-term excessive levels of glucose in the bloodstream has other dangerous effects. It can harm tissues throughout the body, especially those of the blood vessels and nerves. Injury of the lining of arteries lead to atherosclerosis and an increased risk of heart attack and stroke. Damage to the blood vessels of the kidney impairs kidney function and can lead to kidney failure. Damage to blood vessels that serve the eyes can lead to blindness. Blood vessel damage also reduces circulation to the limbs, and nerve damage leads to a loss of sensation, called neuropathy, particularly in the hands and feet. Together, these changes increase the risk of injury, infection, and tissue death, contributing to a high rate of lower leg amputations in people with diabetes. Uncontrolled diabetes can also lead to a dangerous form of metabolic acidosis called ketoacidosis. Deprived of glucose, cells increasingly rely on fat stores for fuel. In a glucose-deficient state, the liver is forced to use an alternative lipid metabolism pathway that results in the increased production of ketone bodies, or ketones, which are acidic. The buildup of ketones in the blood causes ketoacidosis, which, if left untreated, may lead to a life-threatening diabetic coma. Together, these complications make diabetes the 7th leading cause of death in the United States.
How can we help as bioengineers? Better glucose monitoring, such as non-invasive techniques, a breathalyzer, continuous glucose monitoring, Dexcom, and insulin pumps with glucose monitoring.